now, so I'll just quickly say welcome everyone. Thank you for joining the Division of Rheumatology Grand Rounds. A couple of reminders before we start the session. This session is currently being recorded and it will be posted later on our website. There will be a Q&A session at the end, so please add your questions in the Q&A feature and not in the chat box so that it's easier to moderate. And now I'll let Dr. Weisstrom introduce our speaker for today.
Wonderful. Thank you so much, and thank you for everyone joining us. Gives me great pleasure to introduce my friend and colleague, Dr. Renuka Nayak. She received her MD PhD from the University of Pennsylvania and then moved out to UCSF for internal medicine residency. She was on the fast track, so we actually were interns together, and then she finished and continued on as a physician scientist, starting her rheumatology fellowship as I was still trying to finish up internal medicine. And she's been at UCSF since that time as faculty. She's currently an assistant professor and a staff physician at the VA Puget Sound. She is an amazing researcher. She's a PI on 2 grants, both an NIH R01 as well as a VA Merit Award. Both are focused on understanding the microbiome. One is on RA specifically, the other is on response to drug therapy. In addition to being a fantastic scientist, she's known as a wonderful mentor. She's a great clinician and colleague. And I'm really excited to hear her work today. She'll be speaking about the microbiome and rheumatic diseases. So thank you so much for joining us today, Dr. Nayak, and take it away.
Thank you so much, Dr. Weisstrom, for inviting me to share my research with you all today. I'm really excited to be here to talk to you about the impact of the gut microbiome on the treatment of rheumatic disease. Oh man, okay, here we go. So I usually like to start with a little bit of making some definitions because people kind of vary in terms of their use of the word the microbiome, but I kind of define the microbiome as the microbes and their genomes that they bring with them that live in or on an environment. So those microbes may include things like bacteria, which are the kind of the easiest to study right now, but as well as viruses, fungi, and archaea. And then of course, all the genes that they bring with them. And there are all types of microbiomes. As a physician scientist, the one that I care most about is the human microbiome, but even within the human, there are different kinds of microbial communities. There's microbes that live in the gut that reside in predominantly anaerobic conditions, those on the skin that kind of exist in an aerobic environment. So there are researchers that kind of look at different kinds of microbiomes in humans, but there are other microbial communities that reside in other environments as well. So investigators study the ocean microbiome, the soil microbiome. And what we've come to appreciate about these microbes that live in these different environments is that they can have significant impacts on the environments in which they reside. So I also like to start by kind of highlighting why the microbiome is important and kind of a related question is like, what happens when you get rid of it? And sort of developments and technologies have allowed us to study animals that lack a microbiome. So this, what I'm showing you here, is a notobiotic isolator. That's kind of a fancy word for an isolator that allows us to colonize organisms with specific microbes of interest or just to keep them germ-free. And so we can study germ-free animals and compare them to their colonized counterparts. And so then that allows us to ask this question, what happens when you get rid of a microbiome? And what we've learned from those studies is that germ-free animals are nutritionally underdeveloped, which suggests that we need our microbiome to help us harvest energy from and nutrients from the foods that we eat. Germ-free animals have stunted immune systems and are more susceptible to infections. And so we've come to appreciate that the microbiome educates the host immune system. It's a very critical educator. And then also, surprisingly, germ-free animals that have neurologic deficits. They're more anxious, they're more hyperactive, and they have learning and memory deficits. And these are kind of the first 3 sort of functions of the microbiome that were uncovered. But since then, we've kind of uncovered a lot of different ways that the microbiome shapes host physiology. And so because of all these functions that the microbiome provides to the host, researchers like myself consider the microbiome as extending the capabilities of the host genome and therefore our second genome to our own genome. And if we understand more about the microbes and the genes that they bring with them and what they do with those genes, we can develop therapies to advance human health and disease. So new technologies have really revolutionized the study of the microbiome. It used to be that we could only study microbes if we can grow them up on a petri dish And then once we grew them up on a petri dish, we could colonize animals, or, we would have to grow them up in animals. But once we kind of grow them up in a petri dish, we can study them in vitro, or we can look at host microbiome interactions by colonizing germ-free animals with these microbes. And it turns out that we were not very good at colonizing or growing or culturing many different organisms. And so it was really with the development of culture-independent techniques that we learned about the diversity of the microbiome and all these different functions that they bring with them. And so culture-independent techniques are really sequencing-based technologies and analytical chemistry platforms that have allowed us to ask a lot of questions about microbial samples. So we could take a soil sample or a stool sample and perform sequencing or metabolomics on those samples to understand the answers to some of these questions that I've highlighted here below, like what microbes are present. We can look at the 16S rRNA genes that are present in a stool sample, for example. We could do shotgun sequencing to understand the functions that the microbiome is bringing with them to an environment. We can look at RNA sequencing to understand which of these genes are being transcribed into RNA or translated into protein. And these platforms are usually something like LCMS or MALDI-TOF that allow us to look at the proteins that are producing, or the small molecule metabolites. So what are the microbes doing metabolically? And to me, this is kind of an exciting field because while proteins may not be able to traverse the gut barrier into the circulation, because our immune system will definitely not allow that, metabolites can. So in the gut, microbes produce metabolites. They're very small and they can traverse the barrier and go to distal sites like the brain or the joint and affect immune cells in those locations. And so there's actually a number of metabolites that microbes produce, and we know nothing about those metabolites. And so I think this is going to be a really exciting area of investigation in the upcoming years. So kind of central to the research that my lab does is looking at how microbes contribute to variation in treatment response. So I think all of us as rheumatologists have had the experience of patients in our clinic starting a therapy on them and having this conversation with them, like, we don't know how you're going to respond to this drug. Some of you will have a good response, some of you will have a moderate response, and some of you may not respond at all or have side effects. And so for the drug that I'm going to talk to you about today, methotrexate, this is a common conversation that I have with my rheumatoid arthritis patients. And of course, for the focus on methotrexate and rheumatoid arthritis, a number of investigators have looked at what are the factors that might contribute to this variation response. And investigators have looked at genetics, environmental factors like smoking, diet, other drugs, and the state of the immune system. But what I'm going to share with you today is that I think the microbiome plays a role in contributing to variation in treatment response in our rheumatology patients. And this is not unique to rheumatology. Other clinical disciplines also have found that the microbiome contributes to variation in treatment response. And it could be in as diverse treatments as HIV prophylaxis or cancer immunotherapy. And so, how is it that the microbiome can contribute to variation in how patients respond to a drug? And there's two really facets that I want to highlight today. One is that the microbiome is a metabolic powerhouse. And then the second is that our microbiome is a potent immunomodulatory organ. So for the first, I mentioned that microbes have a lot of genes and enzymes that they bring with them. It turns out that they have 150-fold more genes and enzymes encoded in their microbiome than in our own genome. And so those enzymes not only help us kind of digest food and harvest nutrients from food, but can act on the drugs that we give to our patients. So kind of the classic example of this is sulfasalazine, right? Sulfasalazine is a prodrug. It gets metabolized into its active metabolites by the gut microbiome. It was designed that way. But the microbiome can actually also turn active metabolites like digoxin, for example, into inactive versions of digoxin, dihydrodigoxin. Or a microbiome has been found to take drugs and turn them into toxic metabolites. So the classic example of that is irinotecan, which is a cancer therapy. The liver will detoxify it, our liver, but then when it gets excreted in the bile into the gut, the microorganisms will reverse that detoxifying transformation and make the metabolite toxic again, will make irinotecan toxic again, and that's what leads to chemotherapy-induced diarrhea. So that's kind of one mechanism by which microbes can influence drug responses, by metabolizing the drugs that we give to our patients. But another sort of mechanism is that microbes modulate the host immune system, and they do so in a number of different ways. So and because of that, drugs may act on microbes to modulate the host immune system. So the mechanisms by which microbes can modulate the host immune system is they produce these small molecule metabolites like short chain fatty acids or ATPs, indoles, and succinates, or they have proteins that are part of their cell wall like LPS, polysaccharide A, or flagellin that can cause immune activation. These microbes may also produce proteins that mimic our own proteins. And so a normal immune response targeting microbes may then result in cross-reactivity with autoantigens. And that's one of the ways that microbes may contribute to autoimmunity. Or microbes may translocate into the circulation and cause immune phenomenon in distal sites. And so that's more likely to occur when the epithelial barrier is breached, or what some people call leaky gut. And so these are just a few of the ways that microbes may modulate the immune system. I think we're also going to uncover other mechanisms by which microbes modulate the immune system. But if drugs act on microbes to, have any downstream effects, that's another way that microbes may modify treatment response. So as I mentioned, I'm going to talk to you today about my research I've focused mostly on rheumatoid arthritis, and I don't need to, explain what that disease is to this audience. But I like to point out to other audiences that it's, not just doesn't affect just the joints, but can affect other organs as well. And it's first-line therapy. Sorry, methotrexate is first-line therapy for rheumatoid arthritis. So every new RA patient that comes in, I prescribe methotrexate for the most part. And I have this conversation that 1/3 of patients will respond, but 2/3 of patients may not have an adequate response and may either need to switch therapy or have additional therapies added on. And when I first started my line of investigation into this, when I was having these conversations in clinic and I was starting my career in the microbiome research field, I had this question about whether this variation is due to the microbiome, and in particular due to metabolism of methotrexate. And I thought that that was an attractive hypothesis to pursue because methotrexate is an anti-metabolite, and it looks just like folic acid or folates. And folate is this critical pathway in cells across all domains of life. So plants need folate, bacteria need folate, we need folate. And folate is kind of reduced by this enzyme dihydrofolate reductase into these tetrahydrofolates that are critical for key cellular processes like purine synthesis, cell cycle, and protein synthesis. And methotrexate was originally designed to target this enzyme dihydrofolate reductase. But because this is such a conserved pathway from bacteria to humans, I was curious, well, do bacteria have enzymes to digest folate? And do those same enzymes also act on methotrexate? So that was the sort of motivation for pursuing this line of investigation when I first started these studies. So then I had to go back to the literature and find out, well, what was known about methotrexate and drug microbiota interactions in rheumatoid arthritis at the time. And most of the studies were really focused on how the microbiome contributes to pathogenesis of rheumatoid arthritis. And I kind of like to think about all of my research and other people's research as, patient studies versus animal studies versus in vitro studies. And I like all three systems because animal models really allow you to get at causal mechanisms, but patient studies really hammer in on the clinical relevance, right, of what's found in animal models. And so I like to think about these different model systems and what do we see over and over again in different model systems. So what was known at the time was that the microbiome of RA patients differs from that of healthy controls, and the immune system of rheumatoid arthritis patients responds to gut microbes. So Prevotella copri has kind of come up as an organism in studies, some studies, but not in others, as being enriched in RA patient microbiomes. Alan Steer's group found that RA patients produce IgG and IgA reactivity to Prevotella copri. So it's been kind of implicated in the pathogenesis of rheumatoid arthritis. There are other organisms as well now that have been implicated in the pathogenesis of rheumatoid arthritis. And then animal models. So if we take an animal model of RA, where the mice develop rheumatoid arthritis or a rheumatoid arthritis-like disease, and we take those animals and try to rederive them or raise them in a germ-free environment, they don't get arthritis. And so then again, that kind of implies that there's a causal role for the microbiome in developing arthritis. And then if you take those germ-free mice and then colonize them with an RA patient fecal microbiota transplant, so you put the RA microbiome into those mice get arthritis. So again, that's implicating the RA microbiome in pathogenesis. So there were a number of studies that had implicated the microbiome in pathogenesis, but not so much in treatment. And what we knew at that time when I first started these studies was that if you take RA patients and you successfully treat them, their microbiome reverts to resemble that of a healthy control. But nobody had really looked at or extensively looked at metabolism. And so then I wanted to know, well, what do we know about methotrexate metabolism? And so here I'm showing you actually the structure of methotrexate, and here's the structure of folic acid. And you can see that they're very similar, right? There's that double ring, pyrimidine ring, and then this benzoate, and then the green is the glutamate. And folic acid has all of those. The only difference are kind of these two groups here on methotrexate. It's not present on folic acid. So there's only two differences between folic acid and methotrexate. And so therefore, both of these compounds actually undergo the following metabolic transformations. So in humans, we have two enzymes, FPGS and aldehyde oxidase. So this enzyme will add additional glutamates, that's what's indicated here by this little subscript N, to convert methotrexate into a polyglutamated methotrexate. And that's actually the active form of the drug. I didn't know that at the time until I started studying methotrexate more. But this is the active form of the drug. Methotrexate is a prodrug, and this works better at inhibiting dihydrofolate as well as other enzymes. And it's also better retained in cells because the glutamates are added inside the cell. It adds all this positive charge, and it makes it hard for the methotrexate to get back out. That's also true for folate, right? So it's a way to sequester folate inside cells. Aldehyde oxidase is an enzyme that's found in the liver as well as other cells as well, but it adds this hydroxyl moiety. It makes methotrexate less active. And again, humans have both of these enzymes. There's a third transformation that prior to our research, the investigators had found that soil bacteria can actually cleave off this glutamate, the green, and use that as an energy source. And so what's left behind is something called DAMPA, and that's not active at all. And the enzyme that does this in soil bacteria is called CpG2. So nobody had really found whether human gut bacteria do this. There were some thoughts in the 1960s that human gut bacteria could do this, but no, all that they kind of saw was that germ-free mice don't metabolize methotrexate as much as colonized mice. And so then it's not clear, is that because of host enzymes or is that because of the microbiome? That was not sorted out back then. And then in the 1980s, these investigators specifically tested human microbes, but were not able to find that the microbes could produce DAMPA, right? So, and then nobody asked any more questions after that. And I decided I wanted to revisit this line of questioning because of all the new technologies that had been developed to look at the microbiome and drug interactions. And so today I'm going to tell you kind of these two stories. One, looking at how do human gut bacteria first of all, do human gut bacteria metabolize methotrexate? And does that affect clinical response? So what I lovingly call, do bugs affect drugs? And then actually out of that line of questioning came this other line of questioning. So I was incubating drugs like methotrexate with bacteria, and then I saw that the methotrexate was actually doing things to the bacteria, which I was not expecting, right? Because methotrexate, we use that in humans. There's another drug like Bactrim that we use to inhibit the folate pathway of bacteria. I was just not expecting methotrexate to act on microbes, but it turns out that it does. And so, that led to this question of, does the gut microbiome mediate some of the therapeutic effects of methotrexate? So do drugs affect bugs and then have downstream consequences on the host immune system? And so both of these questions are really part of my lab's long-term goal to understand microbial mechanisms that impact treatment response to rheumatologic medications. If we understand that, we can, tailor therapy and advance precision medicine for patients with rheumatic disease. Okay, so jumping right into the data now. So one of the first experiments that I did, which was very exciting in the lab, was just a simple microbiology experiment in vitro where I took bacteria, grew them up on a petri dish, and then grew them up in liquid media, So here's Clostridium aspergiforme. So she's just one microorganism growing up in liquid media, incubated it with methotrexate, and then looked at multiple time points from 15 hours to 168 hours, and then used this platform called HPLC to look at what's happening to methotrexate. So methotrexate here is this black peak. And over time, what you can see is that that is decreasing. And there's this other peak that's increasing over time. And so this data was suggested that C. aspergiforme can metabolize or transform methotrexate, okay? And so that's kind of as far as we can go with HPLC. I don't know what that compound is, okay? But we can kind of do this for multiple organisms, and that's what we did. We looked at 45 different gut species that are found in the human gut. We selected them because they're representative, and they span kind of 6 different phyla, which are the big categories of microbes that exist in the gut. Some of you may be familiar with Bacteroides and Firmicutes because those are sometimes in the media. But when we looked at 40 different gut species, we found that the ones that are highlighted in red can transform or metabolize methotrexate. And what you can see here too is that most of them kind of reside in this one phylum, the Firmicutes, except for this one, Delphtia. And that's actually not a gut bacteria, that's a zoobacterium. That was our positive control, right? That people in the 1960s or whatever had found that zoobacteria can metabolize methotrexate. We also saw that in our assay. So that was in vitro, and that was really one of the first experiments that I did that was super exciting. It immediately led me to ask what happens in patients. And so for this, I collaborated with Jose Scher, who had been collecting stool samples from microbiome RA patients for a lot longer than I had at that time, and working with Andrew Patterson that had access to advanced analytical chemistry platforms like LCMS, which that platform allows us to actually determine which metabolites are being produced. We studied, along with Maggie Alexander, a postdoc in the lab, we looked at RA patients before they were treated. So these are treatment naive RA patients, their stool samples, and we incubated those stool samples with methotrexate, okay? And we followed those patients, or Jose had followed those patients, and had determined whether those ended up being responders or non-responders. But we were working with their pre-treatment microbiome, okay? And in these ex vivo studies, so like now outside of the human, in a petri dish or in a test tube, we incubated those microbial communities. So now these are complex microbial communities with lots of different organisms with methotrexate, and then looked at what happened to methotrexate over 48 hours. And what we can see is that some microbial communities from some of these patients quickly metabolize the drug, and other microbial communities really don't. There's a lot of variation across people's microbiomes as to whether they metabolize methotrexate or not. And then when we overlay whether those patients are responders or non-responders, we tend to see that non-responders quickly metabolize the drug. And so this is just another way to depict that same information, that if we look at the patients and their DAS28 response at 4 months, people who had a better response, which are kind of out here, they had less elimination of methotrexate in this assay. Their microbiomes had less elimination of methotrexate, whereas the patients, each circle here represents a patient's microbiome. Each patient that had microbiomes that quickly metabolized the drug had less of a response. So this was cool because it showed that ex vivo metabolism of methotrexate was associated with clinical response, right? And so that led us then to look in vivo. So this is ex vivo. It was, we haven't actually looked in the blood of these patients yet. We will, but I don't have that data ready to show. It's not ready for prime time. But what we did was then we looked in mice, where we can control for everything, right? We can control for diet, we can control for housing, we can control for genetics, and look what happens to methotrexate in mice that are colonized with different microbiomes, or germ-free versus colonized. So in this study, at this time, we knew that methotrexate was metabolized into DAMPA. What we had also discovered with Andrew Patterson is that there are these novel metabolites. So DAMPA is removal of this glutamate, But DAMPA can also be further metabolized, or maybe this comes from the original methotrexate. These new novel metabolites, 6-MPD and methylpaba, were also seen in our studies using these newer platforms of analytical chemistry, right? So now what we did was we took germ-free mice and we compared them to mice that were colonized, and we treated both groups of mice with oral methotrexate. And so our hypothesis was that, well, germ-free mice really shouldn't produce these metabolites if they're exclusively microbially produced, but colonized mice should. And germ-free mice, if they're not metabolizing methotrexate, should have higher levels of that drug in the blood. And so when we looked using LC-MS at plasma methotrexate levels, that's what we found, that germ-free mice had higher peak methotrexate plasma levels as compared to colonized counterparts. And that was significant. This is unpublished data now. And then that was looking at methotrexate. When we looked at these metabolites, we didn't actually have a method yet developed for this metabolite, but when we looked at the metabolites, we saw that the metabolites were only seen in the mice that were colonized and not in the mice that were germ-free. And we're looking in the plasma, right? So that means that like the methotrexate this is oral methotrexate, was metabolized by gut bacteria, and the metabolites are entering the circulation. And so that was exciting to see this kind of proof of principle, right? In these kind of contrived settings, right? We don't have germ-free humans. But so then the next question we asked was, well, what if we take microbiomes from RA patients and we colonize mice, right? Do a fecal microbiota transplant with different donors? Do we see a difference, right? So does the microbiome contribute to methotrexate pharmacokinetics in vivo in mice? And so for this experiment, we took germ-free mice that were then colonized with 2 different RA donors. So there are 11 mice in each group. And again, treated them with 1 dose of oral methotrexate and looked at plasma pharmacokinetics. And what we can see is that, the donor status or the microbiome colonization status determines the PK profile of this dose of methotrexate. And this is over 8 hours. And so that's exciting because it suggests that some of those things that we were seeing ex vivo, right, in patients, this variation is translated into this in vivo model. And so obviously the next step is to look at what's happening in patients. So one of the questions that I get asked a lot about is, well, you're doing these studies with oral methotrexate, the gut microbiome is metabolizing the drug. Can we bypass all of this by giving subcutaneous methotrexate, right? Which is an option in our patients. Most of my patients don't want to start with subcutaneous methotrexate. They prefer to start with oral methotrexate. But I know our pediatric colleagues usually start with subcutaneous methotrexate. Maybe we could just avoid this whole problem by doing subcutaneous methotrexate. And so we wanted to look at that in vivo in our mouse model. And so in this experiment now, we have mice that are colonized, but we treated them either with oral versus intraperitoneal methotrexate. That's kind of our equivalent of subcutaneous methotrexate in the mice. And then asked, do we see these metabolites? So if we can bypass all of this, I guess what we would predict is that we shouldn't see any metabolites in the intraperitoneal treated mice. So when we looked at that, we actually saw that when we look at methotrexate, right, in the mice that got intraperitoneal methotrexate, the amount of methotrexate is higher. And so that's kind of as expected because the bioavailability of both subcutaneous methotrexate and intraperitoneal methotrexate is higher than oral methotrexate. So this result was as expected, right? There's higher levels of methotrexate in the IP treated group. I think what was not expected was that we saw metabolites even in the mice that got intraperitoneal injection. So here's DAMPA. So what we see is that in orally treated mice, DAMPA shows up in the blood at 2 hours, and there isn't any in the IP treated mice. But then at 4 hours, we see DAMPA showing up in the blood. So turns out we can't avoid this problem by giving injection methotrexate. And the reason is because methotrexate is anaerohepatically circulated. So even if you give methotrexate, in this case, intraperitoneally, it's taken up by the peritoneal vasculature, but then, gets excreted into bile and gets into the gut and gets metabolized in the gut and gets absorbed again in the gut. And so, even non-oral formulations of methotrexate will encounter the microbiome, and the microbiome can affect methotrexate pharmacokinetics when giving non-oral methotrexate. So this is the first story. And so what I have shown you for the first part of this story is that microbes, human gut microbes can metabolize methotrexate directly, and that was done in our in vitro experiments. I also showed you that complex microbial communities from rheumatoid arthritis patients can metabolize methotrexate, and there's variation across patients' microbiomes, and that variation is associated with clinical response in these ex vivo studies, and that in vivo, in our mouse models, the colonization state matters in terms of pharmacokinetics of methotrexate in vivo in the blood, right? And so this was the germ-free versus colonized, but now we've done this experiment as well in RA patients, different microbiome donors. And so, you know, this first part of the story shows that gut bacteria metabolize methotrexate and contribute to variation in drug response. And we looked at 3 different model systems, in vitro, ex vivo, and in vivo, and we're starting to get that patient data now as well, but there's still a lot that we don't know. We don't know which microbes have metabolized methotrexate. We don't know which genes are implicated yet, and we don't know whether diet can be used to modify the microbiome to reduce methotrexate metabolism. And I think these are questions that are important to answer because of the clinical implications that you know, one of the things is if we know the pretreatment microbiome is associated with methotrexate metabolism, maybe we can make a predictor about who might need either subcutaneous methotrexate or some other therapy at the outset. So that would be, that's something that we're interested in developing. Patients ask all the time about whether diet can either affect their disease or improve their response to the drugs that we give them. So if we understand this relationship, you know, we might be able to recommend specific diets. And if we understand which genes, microbial genes are implicated, we could either develop drugs targeting those genes or use CRISPR technologies to edit out those genes. And so that was the first story. And as I mentioned, when I was incubating methotrexate with these microbes, what I saw was that the microbes had reduced growth sometimes, not always, but sometimes. And so that's what led to now this second story. And so, you know, I think about our research as having this yin-yang reciprocal interactions, that microbes act on drugs to affect drug pharmacology, but that drugs can act on microbes to affect host phenotypes, like the state of the immune system. So, so now going back to the literature, like what was known before I started all of this about how methotrexate, you know, acts on bacteria. So people had, in the early days of methotrexate, actually, you know, back in the day, people developed drugs without knowing what the targets were. And so that was true for methotrexate. It was known that it targets the folate pathway, but nobody knew about dihydrofolate reductase. It was really fun to read about the history of the discovery of DHFR. Which was made possible by, you know, having a drug like methotrexate. But researchers have looked at bacterial lysates, so cells that have been broken apart, and found that bacterial DHFR can bind to methotrexate, okay? But if you have an intact cell like E. coli, the E. coli, if you incubate it with methotrexate, the E. coli have efflux pumps that quickly remove the methotrexate from inside the cell. So E. coli are really resistant to methotrexate, but nobody really studied whether other human gut bacteria are similarly resistant or other bacteria in general. And so I had the opportunity to look at that question in these studies. And so here I'm showing you what is called a minimal inhibitory concentration curve, or MIC curve, where I incubated a microbe like C. asparagiforme with different concentrations of methotrexate, so 10 different concentrations of methotrexate, and then looked at the growth of C. asparagiforme relative to the control, so, you know, without the drug. And so here you can see that C. asparagiforme grows pretty well in the presence of different concentrations of methotrexate. Maybe there's a little drop off right at the highest concentration of methotrexate. In contrast, Bacteroides theta iota omicron, which is a human gut microbe, with increasing concentrations of methotrexate, you have less and less growth. And when you hit this concentration, 112 micrograms per mil, you have complete growth inhibition, right? So we call that the minimum inhibitory concentration, or the MIC. And then here's Bacteroides feingoldii. So that it's even more sensitive. It has a lower amount of methotrexate that's required to completely inhibit its growth, right? So MIC is actually something we see a lot in the clinical setting. Whenever we send off our cultures and we get susceptibility testing, what they're reporting back out is the MIC. What's the minimum amount of the antibiotic you need to completely inhibit growth? And so one point I want to make here is that lower amounts of drug means that the microbes are more sensitive, right? If you need less drug to kill the microbe, it's more sensitive. So we did this again with these 45 different isolates and saw that here I'm showing the MIC as a heat map, that some microorganisms are very resistant, like C. aspergiforme, where there's no minimal inhibitory concentration, and then other microbes are pretty sensitive. And those tend to be from the Bacteroides phylum, right? And so here's just another way of showing that data, that the Bacteroides tend to have lower MICs, are more sensitive relative to the other phyla. And again, these are big categories of microbes that encompass multiple species: Bacteroides, Firmicutes, Actinobacteria, and Proteobacteria. Now, another common question that I get here is like, well, what is the concentration of methotrexate in the gut, right? Are these physiological concentrations? It turns out we don't actually know what the concentration of methotrexate in the gut is, because everybody's, amount of volume in there, their transit times, like the absorption rate, is variable. But pharmacologists kind of have a back of the envelope calculation. And when we use those kind of back of the envelope calculations, the predicted concentration of methotrexate in the gut ranges from 7 to 56 micrograms per mil. And in that setting, about 33% of these microorganisms would show kind of complete growth inhibition in response to methotrexate. So, this in vitro study kind of raises the possibility that there's biological possibility that methotrexate may be acting on the gut microbiome. So again, that was in vitro. We then turned to our mouse experiments to look in vivo. If you give mice that are, we started with germ-free mice, we colonized them with a healthy human microbiome. We treated them either with saline, low dose, or high dose oral methotrexate. And then we were asking what happens to the microbiome in vivo. So we treated these mice daily and collected stool daily, and then performed 16S sequencing that allows us to tell which microbes are present. And here I'm showing you what is called a principal components analysis plot. There's 5 of them here. They are data reductionality visualizations of high dimensional data. So each microbiome in each mouse probably has 100 different microbes there, but we can't visualize, 100 different microbes for 9 mice. And so these principal components analysis plots allow us to kind of reduce that data into a 2-dimensional plot. And each circle here represents a mouse and its microbiome. And circles, if they're close together, means the microbiomes are similar. If they're farther apart, means that they're more different. And what we can see at baseline is the microbiomes of these mice that were given either saline, low dose, or high dose methotrexate. Before they got treatment, the microbiomes are not significantly different. But after just 1 day of high dose methotrexate, the microbiomes of these 3 mice significantly differ from the saline and low dose treated mice. And with multiple days of treatment, now we also see the low dose treated mice partitioning away from the saline treated mice. And there's still significant differences in the microbiomes of mice that are given high dose methotrexate. So that's kind of like an aerial view, right, of what's happening to the microbiome. But when you start drilling down into phylum level changes and species level changes, what we start to see is that at the phylum level, this, again, big category of microbes, that Bacteroides are sensitive to the high dose methotrexate, and that they're decreasing over this 4-day time period. And that recapitulates what I showed you in the in vitro studies, that Bacteroides tend to be sensitive in vitro. Now we're also seeing it in vivo, okay? So then that was in mice. We now turn to our patient studies. Again, this is a collaboration with Jose Scher, where we looked at rheumatoid arthritis patients. And initially I kind of had this naive idea that, like, well, we should look at all of them. And when we give methotrexate, there's going to be a big shift in the microbiome. And that's not what we saw. That when you group all of RA patients together and look at the microbiome before and after methotrexate treatment, there's no consistent change. And so that was initially disappointing. But then I remembered that everybody's microbiome is different, and therefore everybody's response to methotrexate may differ. So maybe it's unrealistic, right, to have this expectation. But I also remembered, like, some patients are responders and others are not. And so when we stratified patients based on responders and non-responders, what we saw is that there were some consistent changes. And in particular, the Bacteroides phylum showed consistent changes among responders. So almost all of the responders in our study showed a decrease in Bacteroides, except for that one patient in yellow, whereas the non-responders were kind of all over the map. And that's kind of shown slightly differently here. So now we're seeing that a change in Bacteroides relative abundance is associated with clinical response. And that was cool. But it was also cool that like we're seeing Bacteroides come up again, right? So in vitro, in mice, in vivo, and now in our clinical samples. And that's really what then prompted this next study, where we did a fecal microbiota transplant into mice. And so what we did was we took microbiome stool samples from one patient before that patient got treated with methotrexate, and then also after that patient got treated with methotrexate. We took mice, there were probably, like 10 mice in each of these groups. We colonized mice either with a pre-methotrexate microbiome or a post-methotrexate microbiome. And we did an experiment in a gnotobiotic isolator. We didn't have an arthritis model at that time. What we had was a DSS colitis model. So that was our inflammatory challenge. And then we looked at what happened to different immune cell populations in the setting of inflammation, and with this difference in microbiome status, whether the microbiome was exposed or not to methotrexate. And when we looked at these different immune cell populations that have been implicated in rheumatoid arthritis, and we looked at these 3 different immune sites, the spleen, the small intestine, and the colon, what I'm summarizing for you in this heat map is that there were multiple immune cell populations that are highlighted in blue with a little asterisk that were reduced in the spleen, in the small intestine, and in the colon, particularly in the mice that had a post-methotrexate microbiome. So what this means is that mice that were colonized with microbes that had been exposed to methotrexate, the methotrexate had reconfigured the microbiome. And what we were seeing in this DSS colitis model is that there was less immune activation, right? So this is kind of the study that supports the hypothesis that methotrexate is acting on the microbiota to affect the host immune system. And that was cool because suggests that methotrexate not just acts on the host, but can act on the microbiome as well to alleviate inflammation. So that led to then us looking at whether we can recapitulate that in mouse models. So here we're now starting to look at arthritis models. In all the previous mouse experiments I showed you, there was no arthritis. So, the effects that we were seeing were really due to the drug and not due to the disease. Now we're starting to look at disease models. And the one that we chose was an SKG mouse model of arthritis because the SKG model has a couple of favorable properties. It's a model that is known to be dependent on the microbiome, and it's also one that is responsive to methotrexate. And we confirm that in our hands that if you take SKG mice that have RA-like arthritis and you treat them with methotrexate, that methotrexate tends to alleviate their disease. And interestingly, in this mouse model, there's variation in how the mice are responding to methotrexate, as well as variation in their disease. And there isn't like complete inhibition either of arthritis or complete alleviation of arthritis. So then we asked, well, here's methotrexate, it's targeting the host, but probably is also targeting the microbiome. What if we just target the microbiome in theory with broad spectrum antibiotics? So this is ampicillin, vancomycin, neomycin, and metronidazole. And what we see that when we put these antibiotics in the drinking water, the arthritis of the mice that get the antibiotics also, there's an alleviation of arthritis using antibiotics. And this was then supportive, right? That we could, target the microbiome to alleviate arthritis in this mouse model. What we did was then we took the blood of these mice and performed an O-link panel analysis to look at 95 different immune-related proteins, because we wanted to know, are the immune pathways that are modulated by methotrexate and leading to reduced inflammation, are those the same or different than when you treat with antibiotics? And so the antibiotic here, we did it with both females and males. The methotrexate was just in females. And what we see is that, the immune proteins that are significantly different in the methotrexate treated mice, that's different than in the antibiotic treated mice, right? So there's not overlapping pathways in terms of the immune pathways that are targeted by methotrexate or antibiotics. And to me, that suggests that, we should look at the microbiome and find perhaps novel therapeutic strategies to alleviate inflammation. And maybe that'll provide us a way to reduce inflammation in RA patients that doesn't necessarily lead to immunosuppression. So that's kind of where that story is now. And so for the second part, what I've shown you is that that in mouse models, methotrexate changes the community composition of the microbiome. We saw this in patients, that there's a reduction in Bacteroides, and that's associated with clinical response. I showed you that if we take these microbiomes that have not or have been exposed to methotrexate, the microbiomes have different abilities to activate the immune system in the state of inflammation, and that specifically targeting microbes can alleviate inflammation. And so, kind of building out this simplistic model here, methotrexate acts not just on the host, but acts on the microbiome as well. And if we can figure out ways to modulate the microbiome, maybe we could alleviate inflammation in patients. And so again, there's a lot of unknowns here: what microbes, which genes, what metabolites. Patients ask about diet. And then those are, if we understand those, we can develop some of these therapies, or, probiotics or fecal transplants. I think people are doing fecal transplants in rheumatoid arthritis, but there's like a clinical trial that's ongoing, but we don't understand, which microbes are important. I think that's still, that space, there's still a lot to uncover. Okay, so now there are these two pieces that I showed you, right? Microbiome acts on methotrexate, methotrexate acts on the microbiota. And so if that's the case, that raises this question of, like, can we use the microbiome to predict response in patients? Can the microbiome be a biomarker? And so working with Jose Scher, we found that the pre-treatment microbiome, so the microbiome of patients before they start on methotrexate, is different, between people who will eventually become a non-responder and people who will eventually become a responder. So even at the beginning, their microbiomes are different. And when we use the microbiome to predict response, future response, the microbiome model performs well with an AUC of 0.84. It performs better than just using clinical or current pharmacogenetic models. And then when you combine them, it kind of performs the same as just the using the microbiome as a biomarker. And so we are actually now repeating this study to see if it replicates. And I think I should have that for you, in the future, that story. So taken all together, I think that what this suggests to me is that there are definitely multiple things that contribute to clinical response to methotrexate in RA patients. Previous researchers have looked at genetic determinants, social determinants, but I think that these studies that I've shown you today suggest that the microbiome matters as well. And I think that methotrexate acts on the microbiome, especially if a patient I think patients that are responders tend to have microbiota that are sensitive to methotrexate. Patients that are non-responders, I hypothesize that those patients likely have or tend to have microbes that are resistant to methotrexate, and that resistance may be conferred by the microbes actually metabolizing the drug, right? And so when they do that, these non-responders may not, the methotrexate won't act on the microbe, and then there won't be any methotrexate to act on the patient as well. So that's, I think, how I'm tying all of this together. Some of these data are already published in this arthritis and rheumatology paper and the cell host microbe paper, and also in an eLife paper by Anna Marie Boussillon. And then we're hoping to get some of this unpublished data out soon this year. So there are still a number of questions that are remaining, but I do want to leave a little bit of time here for Q&A from the audience. But I think that what I want, my take-home message is to say that, the microbiome is an underappreciated source of variation in drug response. And I focused a lot on efficacy today, but I think that these findings raise a lot of questions about toxicity. I think that we should, I think there's some promising data here to suggest that the microbiome can be used to tailor therapy. So right now, everybody gets methotrexate, but I would love to build out these models so that we can decide who gets methotrexate orally or subcutaneous methotrexate or biologics. And then I would love to know which genes or metabolites are implicated from microbes so that we can develop targeted therapies targeting the microbiome instead of the host immune system. Or in conjunction with the host immune system. And so with that, I would love to thank you for your attention. I'd like to thank the members of my lab. Diego performed a lot of these pharmacokinetic experiments in mice, and he's going to be a PhD graduate student at Washington University in St. Louis. this fall. And then Judy Ashuri is a close collaborator here. We worked with her on the SKG studies, and then we took her SKG mice and now we re-derived them in a gnotobiotic isolator setting. So now we can do some of these experiments in gnotobiotic SKG mice to look at these interactions between diet and drugs and the microbiome in these like controlled settings. Obviously the microbiome core facilities here, the gnotobiotics core and also the metabolomics core, and then our collaborators, Peter Turnbow was my mentor and now close collaborator, Jose Scher and Andrew Patterson, and then of course our funding sources. And so with that, I'm happy to answer any questions.
So wonderful. Thank you so much. We've got some questions percolating in the Q&A. I'll just read them out loud so we can kind of stay on track. Does that work for you?
Yeah.
First up says, thank you so much for this fascinating lecture. What is the role of probiotic supplementation in RA? Patients occasionally ask about the use of probiotics. And in light of your talk, should we recommend against probiotics that contain Bacteroides? Do you have any thoughts on that?
So yes, I do also get asked a lot of questions. There's a number of like small studies, meaning like the number of patients that are in the studies are small, randomized controlled studies looking at probiotics in rheumatoid arthritis, and there's promising results there. The problem with probiotics is that there's no standard formulation, right? So these different studies use different microbes. I don't know that there's been any like, cross comparison to see which microbes kind of turn up as being helpful in multiple studies across multiple populations. And then, if you get probiotics at the supermarket, it's not always 100% clear that what they say is on the bottle is actually also what's in the bottle, right? The probiotics are not regulated in the same way that drugs are, right? So the quality may not be there. And also probiotics, the definition of a probiotic is that it's a live microbe that is beneficial to the host. That's kind of the WHO definition of probiotics. And there's also like no demonstration that what's in the bottle is alive and can like confer that beneficial effects. So, that being said, I do have patients that anecdotally will say they've been on a probiotic and that it's helpful to them, and so I don't try to like dissuade them that that's not true. I think that it's hard to answer that question because there's no standard formulation. There are some formulations like VSL3, which is used sometimes in the gastroenterology sphere. But I don't know that that's been shown to be beneficial in rheumatoid arthritis. So then this other second part of the question about what should we recommend probiotics that are not Bacteroides due to sensitivity to methotrexate. I don't even think Bacteroides are commonly found in probiotics because probiotics, those are obviously like, those are exposed to oxygen and a lot of Bacteroides tend to be anaerobic. So we can't make that kind of recommendation now. That being said, like we could ask, one of the things that does change Bacteroides abundance is diet. And so, maybe a related question is, should we recommend diets that do or don't expand Bacteroides? And I'm not ready to put my nickel down on that one either, because Bacteroides is such a big phylum, and there's so many members of that phylum that I don't think we understand, which ones are beneficial, which ones are harmful in the setting of RA. I think I also see like a question about which specific species, are known to worsen or improve RA. And I think that's the level of information that we need to know in order to make specific recommendations to patients. It's the, and there are some, while I'm kind of talking about that question, that's the third one down, there are some species that are implicated in RA pathogenesis in mouse models. I don't know that I've seen human studies, except for Prevotella capri, where there are human studies and mouse studies implicating that Prevotella capri contributes to arthritis. There's Subdogranulum didolexi, which is from Christine Kuhn's research group at University of Colorado, where they found that organism in RA patients and has shown that organism leads to arthritis in mice. There is Actinomycetes Agrobacter actinomycetes comitans. That's another microorganism that has been implicated in mouse models of inflammatory arthritis. I'm not sure that I've seen that one in humans, but there's a really nice review that's coming out from Michael Holler's group that I think will hopefully summarize what we do know about which microbes contribute to RA pathogenesis. So I think that's question number one and
Yes, you got them. Awesome. And just building a little bit on that, anything about anti-inflammatory or autoimmune diet while we're talking about diet and supplements?
Yeah. So anti-inflammatory diets, there's kind of a number of different diets that have been implicated in reducing inflammation in patients with autoimmune disease. So the one that I kind of like to suggest for my patients is the Mediterranean diet, in part because it like reduces the risk of cardiovascular disease. Some people will say eat diets that are high in fiber so that the microbiome generates short chain fatty acids, which only the microbiome can produce short chain fatty acids from fiber. But I think the bottom line here as well is that we don't know what to tell patients, in part because everybody's microbiome is different. And just like how we saw that drugs act on the microbiome in personalized ways, diet does the same. So there's this whole field called personalized nutrition, where they're looking at how diet interacts with the microbiome and how that differs across people. And so I feel like where we are on very strong footing in both humans and in mice is to say like high fat diets are bad. So get rid of high fat diets, get rid of, like junk food, processed food. And if patients can do that, they're kind of, I feel like confident about making that kind of statement. But like whether to go on Dr. Whale's anti-inflammatory diet or Mediterranean diet or like diets that are free of nightshades, that I feel like we don't have a lot of evidence yet for. And so it's hard to then give specific guidelines about what people should eat.
All right, this is great. You're on a roll. Okay, a really important question from Keith Elcon. When you give methotrexate to the mice, you get changes in the flora and the immune system. How do you know which comes first?
Yeah. How do you know which one comes first? So when you target with methotrexate, which targets both, right? You don't know which comes first. But if you use something like Bactrim or Septra, which has way, 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 way higher specificity for microbes than for humans, right? And if you see there a reduction in arthritis or a reduction in and inflammatory cells, that way you can say that it's like the drug acting on the microbiome. But now we're talking about a different drug, right? And so the other way to get at that question is methotrexate. In the setting of methotrexate acting on the microbiome, the studies that I showed you today where it changes community composition, those were done in non-diseased mice. So it is most likely the case that methotrexate in the non-diseased mice is first acting on the microbiome where the methotrexate is given by oral gavage, and then entering into the circulation and perhaps affecting the immune system. But I think that when you have something that targets both, it's really hard to know which one comes first. Although I guess maybe what we could have done in that study was to profile both the microbiome and the immune system at the same time. We didn't do that in that study. Yeah.
You could try immunodeficient mice.
Yeah.
Okay.
That's a good point.
Building on the Bactrim question, there's an interaction between Bactrim methotrexate. It increases methotrexate toxicity. Is there a possible benefit using the two together?
Yeah, that's a good question too. So we're looking at that, whether there's like synergy. So for example, in vasculitis patients, they're used together, right? Sometimes. And in rheumatoid arthritis patients or in other patients with autoimmune disease, like the theory between Bactrim and methotrexate is that Bactrim reduces renal clearance of methotrexate. But it's also possible, and that's kind of the supposed mechanism of toxicity there. But it's also possible that both of those drugs are toxic because they act, both of them, on the microbiome, and that's like too much for the patient to handle, And so, one of the questions that we're asking in my lab that we're actually funded through the VA for was whether we could use those synergistically at lower levels, right? So if patient is like having side effects from methotrexate, can we kind of use lower doses of methotrexate with like a little bit of, Bactrim and then get that effect on the microbiome to reduce inflammation? I think it's an interesting question that we're looking at. It also makes me a little bit worried about like, are we inducing resistance? Are there going to be some of the side effects of chronic antibiotic therapy? But then I feel a little bit better knowing that like that's what we do in vasculitis patients, but also vasculitis patients have, infectious complications. I think there's a fine line there, but it is definitely an interesting question and one that we're looking at.
I feel bad I'm going to cut in front of Mark Wenner with my question. So there's new data now to support finally the split dosing, same day split dosing of methotrexate. What do you think is going on from microbiome or for those people to increase their clinical response?
Yeah. So, I don't remember in that paper whether it's like a reduction in toxicity. So maybe like if there's a microbiome that's contributed to toxicity, right? When you split the dose, you have less methotrexate to like have like a toxic effect. And so maybe patients are then more adherent and then more tolerable. And so that's one possibility. But also if it's a lower dose, right? It's possible that the microbiome may be metabolizing the drug. But I do think that I think that the patients that are responding may be ones that don't have a lot of metabolizing microbes, and so maybe the thing is that you're just reducing toxicity and increasing bioavailability. But I don't know. I don't know the answer to that question, but it's definitely
I'm going to ask this last question. I know we're at the hour, but we're just bleeding into our time. So if you're going to stick around, stick around. Mark Wenner asked a question. Thank you for this talk. Have you looked at dose response of microbiome to methotrexate measured by dose of methotrexate and methotrexate plasma levels? What about DAMPA versus native methotrexate plasma concentrations in RA patients in association with microbiome?
Yeah. So dose response.
Sorry, I put in the answer.
That one was like a long one, so I just want to make sure I didn't miss any of it.
Click in the
so
middle.
measure dose response and methotrexate plasma. We haven't done that yet. We should. And then what about DAMPA versus native methotrexate plasma concentrations in RA patients? Is there an association with microbiome? We're looking at that question now. So, and we're looking not just at DAMPA, but some of the other metabolites as well, looking in the stool. We have some urine sample as well, because some of these metabolites may be excreted in the urine. And then we have plasma samples from patients. I don't know the answers to that question, but I hopefully will in the future. Thank you. Thank you for these great questions.
Yeah, my last question is about just the composition of the cohort. And if people need to go, I'm just going to pick Renuka's brain real quick. Do you feel like this is a pretty heterogeneous? Is this like all very seropositive group you're dealing with, or are there seroneg people in there? Do they have similar disease duration, et cetera? And are they like new methotrexate starts?
They're all new methotrexate starts. That's kind of like a requirement for the studies that we've done so far. And so obviously if they're new methotrexate starts, we're not going to find any of the metabolites or methotrexate, like in theory we shouldn't, but we're following them longitudinally. They are patients that are seropositive, so they meet the ACR criteria for rheumatoid arthritis for inclusion into research studies. We haven't looked at seronegative patients, but when we enroll patients in the clinic, we're kind of have a broad net, because when a patient comes in and they have inflammatory arthritis, sometimes you don't know, is that RA, is that psoriatic arthritis? Right. You have to follow them longitudinally. So we're building out our stool collection repository here at UCSF and at NYU. And, the other thing that we're doing is with our stool banks, we're harvesting microbes from the RA patient microbiomes to look at these questions of which specific microbes are metabolizing, what are the metabolites that they're producing, are those metabolites associated with efficacy or toxicity? But yeah, they're actually really hard population to recruit, because the patients are coming new onset, so much is happening. And sometimes like the last thing they want to do is like, submit a stool sample for research.
Right. Totally.
But yeah, we're super
It's hard enough getting people after they're stocked in with their disease. So, wow.
Yeah. But yeah, so we try to make it more of a homogeneous group for that reason. Yeah.
Cool. All right. Well, I think people are taken out, leaving the talk. Thank you so much for sharing your research. It's fascinating. You're such a thoughtful scientist. It's wonderful. So
thank you so much for giving me the opportunity to share.